Hello and welcome to the fourth topic on the topic of molecular biology, which will be including proteins. So within this presentation I'll be talking about polypeptides, the building of a protein or proteins, denaturation of proteins, as well as the functions of proteins. So I hope you enjoy. So polypeptides are built from amino acid monomers during a condensation reaction, and if you are interested in the specifics of that, check out my first video on molecules to metabolism. Polypeptides also become dipeptide when two amino acids are joined with a reaction between the amino group of one carboxyl group of the one and the other, so they form a peptide bond, which then through further condensation becomes a polypeptide. So it's always a simple pattern of a mono one becoming a di something becoming a poly something. And in living cells, proteins are synthesized from ribosomes, which you may remember on the videos of cell biology. If not, check them out. Their proteins are also used for the biosynthesis of proteins, or the creation of protein, which I'll be talking about in one of the next videos. And the sequence of amino acid is coded by the organism's genes, so the so-called codon provides a direct template for amino acid sequences. Exactly, so here you see the structure, the possible structures of a protein. So the primary structure is simply the amino acid or the amino acid chains, and the secondary structure is the formation of hydrogen bonds between the different parts of the chain. So you have two possible shapes depending on amino acid. One, you have the alpha helix and the beta pleated sheets. The tertiary structure is a complex that's three-dimensional in shape and held together by ionic bonds, so between the ions, and disulfide bridges, and it determines where substrates can bind. Lastly, what you cannot see in the picture here is that there is a quaternary structure which has more than two polypeptides. So it's also a 3D structure, but more complicated than that of the tertiary structure. And moving on to denaturation, it destroys the complex structure of proteins through heat and the presence of strong acids or alkalis that disturb the bonds, the hydrogen bonds, no, the peptide bonds between the amino acids. But the primary structure remains, but it loses the biological function and the ability to reconnect again to a higher structure than the primary one. And specifically heat disrupts the hydrogen bonds, as I already said, and the proteins thus unfold due to the high temperature. And generally proteins have in the body, for example, have an optimal temperature of 37 degrees, and if it goes higher than that, denaturation occurs. On the other hand side, we have the pH value, so the acidity or the basicness of those, and amino acids are swiderion, so they have two kinds of charges, and pH alters the charge of the protein, which also leads to unfolding and disruption. And here you can see a folded protein, and through denaturation becomes literally an unfolded protein. The functions are directly linked and determined by the shape of a molecule. So first of all, you have a globular one, which are polypeptide chains folded into spheres. They are hydrophilic and catalytic, and may have, well, as I already said, catalytic functions, as well as regulatory, transport, protective, signal, and transduction ones. We also have, an example for that would be, for example, hemoglobin. We also have fibrous proteins that are parallel polypeptide chains, so they're not really folded into one, and they're cross-linked along the lengths. And those have hydrophobic properties and are physically tough. They have structural roles and contractile roles. And lastly, something that's not really linked to the shape, but in general, we have proteomes. that every individual organism has their own unique proteins that are determined by the unique genome. And that was it on proteins. Thank you for watching. These are my sources. Have a nice day.